Dear students, happy to meet you in this online mode of education. The topic I am going to discuss with you today is about the edible oyster culture. I am Dr.S.ANGELINA PARIMALA, Associate Professor and Head from the PG and Research Department of Zoology, A.D.M. College for Women, Nagapattinam. Let us see something about the introductory part. The edible oysters are highly proteinous food, and it is present in the sea. So it is a major seafood. And in India, there is a growing demand for oyster meat in some parts of the country. It is said that oyster is scientifically the best known marine animal. It is one of the most widely cultivated species. As early as the 1st century of BC, the Romans were the first to develop simple methods of collecting oyster seeds and growing them for food. The Japanese, they developed farming methods that yielded good results. The awareness about the vast potentialities for the development of oyster farming in tropics is more important. And also we can see some serious efforts are now being directed in its development under tropical conditions. When we see the biology of oyster, the shape of the oyster is extremely variable depending on the environment in which it is grown. Smooth and elongated, and also when they are grown individually on the soft substrata, they are beautiful mollusk forms, and also uh, corrugated and circular shell of its with a lower value deep when grown individually on hard substratum, and also it is having the irregular shape when grown with other oysters. Generally, we can see the shape related to a circular, and the shape is related to elongated with reduced cupped nature when grown fixed to a firm substratum. Reproduction of this oysters in the genus Cassiopeia, sexes are separate but occasionally hermaphrodites, and during spawning time, the ripe eggs and sperms are discharged into the exterior where fertilization takes place in the outer surface. That is, external fertilization is taking place. And the temperature, food availability, and salinity are considered as important exogenous factors in influencing the maturation of gonads. A single female measuring 80 to 90 spawns 10 to 15 million eggs at a time. When we see the condition index, here the condition index of the oysters denotes the quality of the meat, and it is useful to determine the best period for harvest. It is also helpful to access the suitability of a locality for culture. And also high condition indicates greater proportion of meat in the whole weight of the oyster. Those in prime condition are tasty when compared to the flaccid and watery meats of oysters in poor condition. The soft body of the oyster undergoes changes which are usually related to the reproductive cycle. During the maturation process, the gonad increases in weight, resulting in increase in the weight of the soft body. Before the commencement of spawning, the condition index reaches high value, and with release of gametes, the soft body of the oyster loses weight. And also, this condition factor is studied based on the weight, volume of both. The percentage of wet flesh weight in total weight gives the condition. When we see the condition index, that is equal to weight of dry meat weight into 1000 bar volume of shell cavity. In Matasensis species, the condition is considered as high if it is above 140 and poor if it is below 70. When we see the seed collection here, the oyster seeds are collected from estuaries by placing suitable collectors called clutch in the water column at appropriate period. During spawning season, the fat collectors are suspended from racks. When we prepare the clutch, and this clutch is the term used for fat or seed collector for suspended method of oyster culture. The clutch made of oyster shells have been found to be ideal, and empty oyster shells are cleaned manually to remove the foulness and then washed to remove slit also. A small hole is made on the shell, and these are strung on 3 mm in size and the diameter, and nylon rope with a spacing of 15 to 20 centimeter between each shell. That is 5 shells per meter rope we can see. And such strings are called wrens. The spaced wrens can be used as for grow these oysters. For seed collection purposes, the shells are strung continuously without spaces. In that case, we can see 10 to 15 shells per meter. And after the attachment of seed, the shells can be removed and restrung at the rate of 5 shells per meter, which is the ideal density for growout. If the oysters are to be grown by the tray method, then empty shells or line-coated tiles can be placed in the trays for seed collection. Line-coated tiles gave encouraging results, and on the single tile, as many as 120 larvae are known to settle. One of the main factors that determine the success of the forming operation is the period when the clutches are placed for seed collection. If they are laid in advance of spatfall, they may be covered with a slit or settlement of fowlers, making them unsuitable for the oyster larvae to settle. The larval period in Madasensis species is 15 to 20 days. The ideal time for laying the peak spawning, and here it is determined by gonad examination and the absence of early larval stages in the plankton. Strong currents interfere with the larval settlement and may result in poor spat collection. When we see the selection of pop sites, here the sheltered areas offering protection from strong wave action are preferred. From intertidal region to areas extending up to about 5 meter depth can be considered for adopting suitable culture methods. Similarly, the culture technique is adopted depending upon the type of substratum also. On bottom culture method is substrate here specific, while off bottom method has little to do with the nature of substratum. When we see the large scale mortalities have been reported in salinities below 10 and above 40 ppt, when the natural oyster populations of particular species, Madrasensis, were exposed for prolonged periods. The natural populations occur at the temperature ranges from 21 to 31 degrees centigrade. When we see the different types of forming methods for these edible oysters, here they are broadly grouped as on bottom culture and off bottom culture, raft culture, rack culture, then long line and stake culture are used in various off bottom culture practices. The off bottom culture methods are advantageous over the bottom culture in the following respects. When we see this, it is relatively having the rapid growth and good meat yield we can get, and also the facilities are three-dimensional utilization of the culture area, and also the biological functions of the oyster such as filtration, feeding, etc. are carried out independent of their tidal flow. and also slitting and predatory problems are negligible. When we see in the on-bottom culture, the oysters are grown either in the intertidal or subtidal area directly on hard substratum. For intertidal culture, a minimum of 16 hours submergence is suggested to ensure adequate food supply. And also oyster feed attached to the collectors are planted on the bottom and allowed to grow for the market. The advantages of this method are increased exposure to benthic predation, siltation, and low production. In USA, the production is estimated at 5 tons per hectare per year, and in France, 7.5 tons per hectare per year. This method is yet to be experienced in India. When we see the rack and wren method here, the nursery related reared single spat culture tree measuring about 25 mm or transferred to trays of size 40 into 40 into 10 centimeters at a density of 150 to 200 oysterlings per tray. The tray is knitted with 2 mm synthetic twine of appropriate mesh and is suspended from rack. Once the oysters reaches 50 mm in length, they are segregated and transferred to rectangular tray of with the size 90 into 60 into 15 centimeters. These trays are placed on the rack. Each tray holds 150 to 200 oysters. The average growth rate of the oyster is 7 mm per month. And at the end of 12 months, the oysters attain an average length of 85 mm in category. The production is estimated at 120 tons per hectare per year. Compared to the stirring method, this method gives production, but the production cost is very high. It is also called the wren method. The racks are constructed in 1 to 2.5 m and depth. There are several variations in the types of racks. The single beam rack consists of a beam placed and secured to the top of posts driven into the bottom. A series of single beams are placed in a row. The cross beam rack is constructed by placing crossbar on top, single post, and 2 long beams are secured on the end of cross beams. In the form, the shell strings are suspended from racks. The mortality, including those fallen, is about 45% when we observe it. And also in the stake culture, here a stake is driven into the substratum, and on the top end 1 nail and on the sides 2 nails are fixed. The nail holds in position a shell with a spat attached. The stakes are placed 60 centimeters apart. In this method, the nursery rearing of spat is carried on the same stack for about 2 months. The spat on the top end of the stake are covered by a piece of valance screen. Once the oysters attain 25 to 30 mm, the valance screen is removed, and in another 10 months, they reach the marketable size. The growth rate of the oysters in this method is the same as that the oysters raised by the stirring method. The production is estimated at 20 tons per hectare per year. And in the farm management, here when we see the farm management is very, very important. Here the periodic checking of the farms is very essential. The main points to be checked are replacement of broken farm structure and resuspending loosened rims which touch the estuarine bottom. High mortality rates have been observed when the rims falls on the ground. To tide over these problems, periodic checking is very, very essential. And also, predators and fowlers are the main reason for the oyster farmers. And also, crabs, fishes, starfish, polychaetes, and gastropods are the main predators for the oysters. Barnacles are a fowler that settles on the wooden structure, trays, and oysters. It completes for food with the oysters. It also increases the weight of the wren, causing damage to the farm structure. and also large scale mortalities of oysters due to the diseases caused by the fungus, Trichinitis marinus, and the protozoan parasites have been reported from the temperate countries. And also the oysters in the natural bed at Tuticorin have been observed to be infected by many microorganisms. And when we see the harvest of oysters here, the oysters are harvested when the condition is very high. And also along in the Kerala harvest is ideal during May in Vembanad and Chetua estuary and during August time and August and also to October in Askalamudi lake. Generally, high condition index is obtained when a gonad is ripe prior to spawning. Harvesting is done manually. When your depression do the activity, here the oysters, like other filter feeding bivalves, accumulate pathogenic organisms in their body. Members of the Salmonella group causes typhoid fever, while coliforms and vibrios cause gastroenteritis. By depression, the bacteria load is brought down to permissible levels, also freezes, sand particles, and slits are removed from the alimentary canal of the oysters. The oysters are placed for 24 hours in cleaning tanks under a flow of filtered seawater. About 10 to 20% of the seawater is continuously replaced. At the end of 12 hours, the water in the tank is drained and the oysters are cleaned by a strong jet of water to remove the accumulated feces. The tanks are again filled with filtered water and the flow is maintained for another 12 hours. Then the tanks are drained and flushed with the jet of filtered seawater. The oysters are held for about 1 hour in 3 ppm chlorinated seawater and then washed once again in filtered seawater before marketing. When we see the identification of some selected species of this edible oyster, here they have modifications in their shapes and walls and also adductor muscle scar and a hinge ligament like that. When we see one of our species here, it is having the irregular shape. Usually it is having the elongated shell, and also when we see the valves here, the left valve deep and the right one is slightly concave. And when we see the adductor muscle scar here, okay, it is a kidney shaped and dark purple in nature. When we see the hinge, it is narrow and elongated. When we see the second species here, it is also elongated and thick. And here we can see the left valve is in the cup-like shape. And here when we see the adductor muscle scar here, it is broad and more or less it is oblong. And here we can see the presence of scar. And also when we see in the hinge here, it is well developed, highly well developed, and it has a deep median groove with lateral elevations. In the third one here we can see the large, roughly found and also thick the shallows in the shell cavity, and the shape is slight irregular. And when we see in the walls here, the left wall is very thick and slightly concave, and in the right side we can see the same size or slightly larger compared with the left side. And in the adductor muscle here, it is oblong and white or smoky white in color. When in the last species here, it is again the shape of the shell. It is a very hard, strong, and when we see the walls, the margin of the both walls have well developed. And also in the adductor muscles here, we can see the cream colored muscles we can observe. And in the hinge ligament here, it is straight and also teeth-like structures we can observe. And here it is also well developed. When we concentrate on the transport and storage, here the oysters kept under moist and cool conditions survive for several days. However, it is desirable that they reach the consumer within 3 days of harvest. Studies indicates that oysters packed in wet gunny bags are safely transported for 20 to 30 hours without mortality and in good condition. When shucking, the removal of the meat from the oyster, that process is called shucking. Here, a stainless steel knife is used for the purpose. To render stacking easy, oysters are subjected to a wide range of treatments such as exposure to weaker hydrochloric acid, heat cold, then vacuum, microwaves and lasers, and also freezing the oysters or immersing them in hot water are the two methods commonly followed. However, in India, steaming the oysters for 5 to 8 minutes has been found to be ideal to make the oysters to open their valves. When we do the processing, here the oysters are eaten in fresh condition in the half shell in many countries. The oysters are processed in several ways. When we see the frozen oysters after preparation, while oysters shell on are frozen by spreading them in a single layer of trays in an air blast freezer with polypropylene film stretched over each tray. Frozen whole oysters packed in polythene bags remain in good condition for 6 months in cold storage at the temperature with 25 degrees centigrade. The liquid within the shell acts as a glaze to protect the meat from dehydration. When we see observe the canned oysters, here the oyster meat is chilled, washed, and blended in 3% brine containing 0.1% citric acid for 4 to 5 minutes. The balanced meat is packed in cans and a hot 2% brine with 0.1% citric acid is added to the cans. The cans are steamed, sterilized at 115 degrees centigrade for 25 minutes and immediately chilled and stored. When we see the smoked oysters, here the meat is washed, treated with 5% brine for 5 minutes, then drained, dipped in edible oil, spread in a single layer on a nylon wire mesh, then drained again and loaded into a smoking chamber. The meat is held in dense smoke and maintained at a temperature of 40 degrees centigrade for 30 minutes and later at 70 degrees centigrade for 90 minutes. The smoked oysters are filled in cans with hot refined oil. The cans are steamed, sterilized at 115 degrees centigrade for 25 minutes and immediately chilled and stored. When we see the oyster stew, this is also one of the unique feature. Here, the oyster which are too large or barely cut while stacking or these in low condition are prepared as for canning but are chopped into small pieces and added to milk and spices. When we see some byproducts and their utilization, the two shell valves constitute about 85% of the total weight of oyster and contain 52 to 55% of calcium oxide. They are used in the manufacture of calcium carbide, lime, fertilizers, and cement. They are useful fat collectors in oyster culture. The shells are broken into pieces and used as poultry grit. When we see the quality of oyster meat and shell, here the oyster meat is released in most nations. The proximate composition of the oyster meat and medicinal values attributed to it. And when we see it, the oyster meat consists of 52% of protein, 14% of glycogen, and 11% of fat. And also when we see the oyster powder, it contains wide ranges of minerals and vitamins and the amino acid taurine which has complex medicinal properties. And also the oyster powder has shown good results in skin care, then numerous heart ailments, blood pressure, liver problems, then curing the arthritis and the rheumatism and diabetes, water retention and premenstrual tension. When we conclude this, the importance of this edible oyster culture, it is a very simple technology which can be easily practiced. There are a few critical factors such as the seed collection and harvesting method which are governed by the biology of the species, which affect the profit of their farming operations. The oysters are grown either in the intertidal or subtidal area directly on a hot substrate. For intertidal culture, a minimum of 16 hours subvented is suggested to ensure adequate food supply. Oyster seed attached to the collectors are planted on the bottom and allowed to grow for the market to get more profit. Thank you.